AP 120, Chapter 3, Topics Nucleus and Mitosis. So, the nucleus, third main component of the cell. What does the nucleus do? Well, it houses our genetic code, our DNA, thing that is the blueprints that make each of us individuals, different from each other. So, the nucleus is the largest organelle in the cell. It has a nuclear envelope, which is a double membrane. So it has two phospholipid bilayers forming the nuclear envelope surrounding and forming the nucleus itself. And within the nucleus are these large openings. These large openings are called nuclear pores. And they're there to allow materials to enter and exit the cell, and especially rather large materials. When we look inside of the nucleus, we see a darkened area, and that darkened area is called the nucleolus. Nucleolus is an organelle. Nucleolus is where the component parts of the ribosome are produced. So the proteins and RNA that make up the ribosome are produced by the nucleolus, put together, and then the components leave through the nuclear pores. Also found within the nucleus is chromatin, all scattered throughout. Chromatin is loosely coiled DNA bound to proteins. So in our nucleus, DNA is not naked. It is bound up to proteins that help to hold it together, organize it, and protect it. And of course, DNA is so critically important because it holds our genetic code. Basically, we have approximately 30,000 genes. Each gene is a small portion of DNA that codes for a specific protein. Genes code for proteins. We need those genes to produce the proteins necessary for us to survive. So, for instance, here is the DNA double helix. As I said, it is bound up with proteins, histone proteins. It coils around them and is packaged into the chromatin, which allows the DNA to be accessible as well as properly organized so it doesn't get tangled up into a knot. And then, when the cell decides to divide, the chromatin condenses down to form a chromosome. And the chromosome, as you can see, looks kind of like an X, and it has a point where the two sides are connected, and that is the centromere of the chromosome. And again, you only see chromosomes when cell division is occurring. So, what is the life of the cell? Well, to start with, most of the cell's life is in what's referred to as interphase. Interphase is when the cell grows and where it produces and performs all of its normal functions. So most of the cell's life is in interphase. Say a cell is just newly formed, that new cell will start off in the G1 phase. G1 phase is a growth cell. When cells are newly produced, they're usually a little smaller than normal. So during the G1 phase, they grow to the size and shape they're supposed to be. Now, many cells reach the end of the G1 phase and then exit the cell cycle because that's it, they're done. They're never going to divide again. So neurons, muscle cells, red blood cells, once they're produced, they're never going to divide again. However, a lot of other cells, especially stem cells, will go on to divide again. So they go from the G1 phase that's that. The restriction point decides cells fate if it's going to leave or not. And then they enter the S phase. So cells that are going to divide again enter the S phase. During the S phase of interphase, the DNA is replicated so that we now have two identical copies of DNA. This is critical because when the cell divides into two, we need a copy of DNA that's complete for each new cell. And then from here, the cell enters the G2 phase. G2 is another growth phase. In this case, the cell is getting much, much larger than normal, so that when it divides, there'll be enough cytoplasm and organelles for two separate cells. So interphase, where the cell spends most of its time, has an initial growth phase to get to the normal size, and then if it's going to divide again, it will go into the S phase to produce more DNA, and then the G2 phase to get extra big. Then there is the mitotic phase, and in the mitotic phase is when mitosis occurs, or cell division. And cell division includes mitosis and cytokinesis. So when a cell divides, it also has to divide the nucleus so that one complete copy of DNA ends up in each of the new cells, and it undergoes cytokinesis, which is the division of the cytoplasm, and when the cell actually separates into two distinct cells. So basically, during the cell cycle, we have one parent cell, one starting cell. It enters interphase, goes through the three phases of interphase, and then it will enter mitosis, where it goes through the phase of prophase, metaphase, anaphase, telophase, and then also cytokinesis, and in this process, then divide into two identical sister cells. So mitosis and cell division will lead to two cells identical to each other and identical to the original parent cell. So interphase, again, what's going on during interphase? We have growth. At the end of G2 phase, we have two copies of DNA, two sets of centrioles, so two centrosomes with their sets of centrioles. See them here and here. Additional organelles, of course, for the two new cells that will eventually form, and the cell is larger than normal. So here's an image of a cell in interphase. Here's its nucleus filled with chromatin. The first phase of mitosis is prophase. In prophase, the chromatin coils down into chromosomes. So the chromatin condenses down, leading to the production of chromosomes. There will be 46 chromosomes in the human. Each chromosome has two sister chromatids. These are identical copies of the DNA. They're attached to each other by the centromere. So two copies of DNA per chromosome. Those copies are called sister chromatids, and they're connected to each other by the centromere. Also, during prophase, the nuclear envelope will disappear, and the nucleus will disappear. The ER will break down, and the nucleolus will also disappear. So here, in this image of prophase, we can see the chromosomes forming, and we start to see the breakdown of the nucleus, and it will continue to break down completely till the chromosomes are released into the cytoplasm. We also see that the centrioles of the centrosomes are now on opposite sides of the cell, and they start producing structures called spindle fibers. These spindle fibers will start to grow out toward the chromosomes. And spindle fibers are made of microtubules. During metaphase, the chromosomes all line up along the middle of the cell. So they all line up along what's called the equatorial plate, which is pretty amazing. Then the spindle fibers will grow out to the chromosomes and attach to the centromeres. So the spindle fibers will stretch from the centrioles of the centrosomes and reach the centromeres of the chromosomes. Next is anaphase. During anaphase, the spindle fibers will now shorten. When they shorten, they will pull on the centromeres and end up separating the chromosomes into two equal parts. So the two sister chromatids end up being separated and pulled in opposite directions. And once they are completely separated, the sister chromatids are now called chromosomes, and they travel to opposite sides of the cell, which then means that the two new cells produced will each have identical copies of DNA. Also during anaphase, cytokinesis begins. So the cell membrane begins to pinch around the center of the cell and start to form a cleavage furrow down into the cell. The final phase of mitosis is telophase. During telophase, the DNA, the chromosomes reach the opposite sides of the cells. The nucleus will begin to reform around those chromosomes. As the nucleus reforms, the chromosomes will start to uncoil and become chromatin again. As the process continues, the nucleolus will reform, the endoplasmic reticulums will reform, the spindle fibers will break down because they're no longer needed, And also we get the cytokinesis. So those cleavage furrows will get deeper and deeper until they meet in the middle, separating the one big parent cell into two new distinct sister cells. And so here's an image of telophase happening. You can see the separating via cytokinesis of the cells, the DNA starting to unravel in each of the two new cells. So the cell cycle starts with the new cell entering interphase, going through G1 phase, and if it's going to divide again from there to S phase, where it makes more DNA, it's a G2 phase where it gets bigger. It will then enter mitosis, starting with prophase, when the nucleus breaks down, chromosomes form, centrioles go to opposite sides of the cell and start producing mitotic spindle or spindle fibers. Then it enters metaphase when the chromosomes line up in the middle and the spindle fibers attach to the centromeres of the chromosomes. And it enters anaphase when the spindle fibers shorten, splitting the chromosomes in half at the centromere, taking one sister chromatid in one direction, the other in the other direction. And then finally, telophase, the DNA makes it to opposite sides of the cell, nucleus begins to reform, chromosomes become chromatin again, and the cleavage furrow forms and leads to cytokinesis and the division of the cell into two new cells. That's the cell cycle, going around and around and around for cells that regularly divide. All right, one amazing thing about humans is we start off as a single cell. A sperm cell from the father with 23 chromosomes fuses with an oocyte or egg cell from the mother with another 23 chromosomes. They fuse to form a fertilized egg cell with 46 chromosomes. And then the egg cell is going to start to divide again and again and again, producing those 75 trillion cells, all very distinct and different from each other. There's lots and lots of different kinds of cells, sebaceous gland cells, epithelial cells, neurons, astrocytes, muscle cells, and so forth. And this process of turning a generic cell, a stem cell, into lots of distinct kinds of cells is known as differentiation. So differentiation when a cell becomes a specific subtype of cell. Now, all these cells have the same DNA, so how is this possible? Well, as I hope you remember from the beginning of chapter 3, it's possible because each cell only uses the DNA it needs to become the kind of cell it's destined to be. So while some genes are activated for all cells, because they're important for all living cells, other genes are only activated for, say, muscles. So muscle genes will only be active in muscle cells, neural genes only in neurons, and so on. And that's how we're able to get all these distinct kinds of cells. Finally, there is this process called apoptosis. Apoptosis is programmed cell death. It is important that in certain situations our cells die and die in a scheduled, programmed manner. So many cells will need to die during the development of the embryo of the person and during growth. So for instance, when we were fetuses inside of the womb, we had webbed fingers. There's webbing between our fingers and toes. As we continue to develop within the womb, those cells that formed those webbings underwent apoptosis, died, and we lost the webbing. And that is it for chapter 3.